Let's go ahead and start up the lecture. Now, we're going to be starting on food as fuel. And so this is going to be an overview of metabolism that we're discussing. Now, metabolism refers to the chemical reactions occurring in the body that break down or build up molecules. Basically, it's your building and breaking molecules in your body. In biological systems, the chemical reactions of metabolism occur in a series of steps. These are going to be referred to as metabolic pathways. More often than not, they're actually going to be in different web sort of structures and different cyclic structures. Okay, and so sometimes I'll be referring to these as like cycles, like a citric acid cycle. Okay, but there's still a pathway. The chemical energy in the body is going to be captured in the nucleotide adenosine triphosphate, or ATP. This is how we usually refer to that sort of energy tally in the body. Now, one of the things I do want to point out here is that ATP is not the only molecule that can work to capture energy. You can use GTP, you can use CTP, you can use all of those. Okay? But we're always going to end up referring to those as ATP. We're just going to ignore the finer point that it could be a GTP. Okay? There's actually a couple points where it will be a GTP, right? We don't care, right? It's just a fine point that you don't need. So, animals are going to extract energy from covalent bonds that can contained in carbohydrates, fats, and proteins. So the first stage of metabolism, your large biomolecules in your food are going to be digested into smaller units. This is typically through hydrolysis reactions. You may have noticed that hydrolysis is like the typical way we break up large biological molecules. And so that's the first thing that's always going to happen. You have to break it up. Okay. You already kind of knew this because the first thing that happens in your process of eating is what? Chewing. So you're already starting the breakup process when you're chewing things, right? And you're in fact starting the chemical breakup process on sugars in your mouth. And then you continuously are going to break it down into finer and finer units until they get into small enough units to enter essentially the citric acid cycle. Okay. Now, polysaccharides are going to be hydrolyzed into monosaccharide units. Triglycerides are going to be broken down to glycerol and fatty acids. Proteins are going to be hydrolyzed into the amino acid units. These smaller hydrolysis products are going to be absorbed through your intestinal wall into your bloodstream. They'll eventually be transported to different tissues for use by the cells. Again, this is a generalization. There is a little bit more complexity to this that we're not going to go into a lot, but you do have processes where you can take larger molecules and move them around and reassemble those larger molecules on the other side. Okay? But for the vast majority of these metabolic products, this is what happens. So in the cells, your hydrolysis products are going to be broken down through the oxidative process. This is going to give you a few common metabolites, which will contain 2 or 3 carbons typically. Metabolites are going to be chemical intermediates formed by enzyme catalyzed reactions in your body. These are some very common metabolites that you would see that are ubiquitous to these processes. So you're going to see a lot of things that are pyruvate and derivatives of pyruvate, where they just have a little bit of difference in the structure from pyruvate. You're going to see a lot of acetyl groups, okay? And that's the acetyl group. Remember that acetyl group that you saw at like the end of a fatty acid? Okay? Or like when we were talking about glycerol, where we had triglycerides, same sort of linkage, right? All right. So as long as the cells have oxygen or are producing energy, 2 carbon acetyl groups are going to be broken down further to carbon dioxide through a series of chemical reactions. This series of chemical reactions is referred to as the citric acid cycle. It's a pathway that actually joins back in on itself as a cycle. Okay. Now the cycle is going to work in conjunction with electron transport and oxidative phosphorylation pathways to produce the following energy molecules, which are the main output energy in this cycle. We have ATP, of course. We have NADH, and then we have FADH2. These right here are the oxidation reduction molecules we had talked about before. These right here are the ATP that we've been talking about as like the main energy source in the body. Okay. Now there is a process to convert these molecules one into another, but we're going to just concentrate on what are we producing. Okay. Now, one of the things I would like to know is the citric acid cycle is an incredibly central part of your metabolism. Okay. So you can kind of think of this as like the wheel that everything kind of joins together on, right? So you're going to notice that a lot of these pathways are going to begin and end, begin or end at things that are common to the citric acid cycle. Okay. And if you went further into biochemistry, you'd actually notice these common metabolites kind of show up just everywhere because they're incredibly important because they're in the citric acid cycle. All right. So this is the overall of how metabolism works here. We have our proteins. We break these down into amino acids, right? And then once these are broken down to amino acids, they're going to be small enough that you can start transporting them across cell membranes. And so they're going to go across cell membranes into the cytoplasm. In the cytoplasm, they oftentimes are going to be processed to pyruvate, but they don't always have to be processed immediately to pyruvate. Some of the amino acids can actually go on into the mitochondria. Have you ever heard the saying the mitochondria is the powerhouse of the cell? Is that like mostly what you know about this? Okay, that's fair. This is why the mitochondria is the powerhouse of the cell. Okay? Because the mitochondria is where the citric acid cycle and electron transport chain are located, right? Interestingly enough, the mitochondria actually has its own DNA. It is actually its own cell, technically. It can't actually survive on its own anymore, but it was a symbiotic organism that has joined us now, okay? And you'd be really shocked about how much of your mood, how much of your emotions, your thought process is actually controlled by the mitochondria. So a lot of what you think is you're actually like missing two major parts of what you think is you. You are a combination of human, mitochondria, and the different bacteria organisms that are in your intestine. That's what your consciousness is actually composed of. Okay? It's about equal parts too, so it's kind of funny. Pretty wild stuff. Now, your citric acid cycle here, this is the primary input. You're going to put all these things in. This is actually where your CO2 and your O2 are used to make CO2. And it's the electron transport chain alongside oxidative phosphorylation that is actually where you'll produce your ATP. Okay? All right, now, quickly, the polysaccharides go to monosaccharides. You do generate a little bit of ATP in the process of glycolysis. Okay? But the majority of that is going to go into the form of pyruvate and then acetyl-CoA. This is coenzyme A, by the way. And this is going to be the acetyl groups that are going to go in the citric acid cycle. Lipids actually have two different parts to them. So when they're broken up, you get the glycerol, and that's the heads, right? And then you get the fatty acid, which are those tails, right? And so those actually both get processed. The glycerol goes to pyruvate. The fatty acid tails actually go directly to acetyl-CoA. Okay? They're broken off as different acetyl groups. And then they'll be fed into the citric acid cycle. Now, again, this can be divided into 4 steps. We have the fuel biomolecules, the stuff you would see on the back of packages, right? You have your sugars, you have your lipids, you have your proteins, don't you? Okay? And then you have hydrolysis products, the things that you're going to make by breaking them apart. Okay? And then you're going to have your common metabolites, and those are going to be things like your pyruvate and acetyl-CoAs. Okay? And then you're going to have your end products, and those are primarily going to be things like water, CO2, ATP, of course, FADH2, NADH, and you'll also get some amines as well. Okay? The amines, for the most part, are actually treated more like waste products, And this is, in fact, why you need to urinate. Urea, urine. Okay. So, chemical reactions that occur in living systems are going to be called biochemical reactions, obviously. Just says exactly what it is, right? Chemical reactions occur in a series. It's called a metabolic pathway. Sugar molecule glucose, this is a 6-carbon molecule, is going to be broken down into 2 molecules of pyruvate, 3 carbons each. And this is going to be through a series of chemical reactions, and that's called glycolysis. Metabolism can be considered 2 parts. We have catabolism and anabolism. One of the things I'd like to note is these names are going to mean exactly what you think they mean. Okay? So lysis, to cut or break, right? Gluco as in glucose, right? So we cut or break glucose in glycolysis. Catabolism, this can refer to chemical reactions in which large molecules are broken down into fewer common metabolites. These reactions tend to be exergonic, i.e. you actually have to pay in some energy to the externals. You actually release energy that you can use a little bit later. Okay. The energy released during the catabolic reactions is going to be captured in your ATP, usually, though it can also be in NADH and FADH too. Now, these catabolism, this is going to be what is used to actually drive the anabolism. Okay? So anabolism is going to refer to the chemical reactions where your metabolites will be combined to form larger molecules. These reactions are going to be endergonic, meaning you pay energy in and make these large molecules. Okay? So think of this as you get paid, you pay in. Right? Mm-hmm.
So anabolism energy is released.
For you. Yeah. For your use. Yeah. And then anabolism, you pay in to make these large biomolecules that you need for other things. Okay? Such as like messengers and proteins, that sort of stuff. Okay. So you can again think of catabolism as taking large biomolecules to smaller molecules and energy. Anabolism, taking small building blocks, going to large molecules, which takes up energy, okay, which expends energy. All right, so again, that's a summary chart. The metabolic pathways are going to occur in different parts of the cell. In animals, the cell membrane is going to separate the materials inside the cell from the exterior aqueous environment. The main structure component of your cell membrane, again, is going to be those phospholipids. The nucleus is going to contain your DNA. This is going to control things like cell replication, and it's also going to control the protein synthesis for that cell. The cytoplasm is going to consist of all the material between the nucleus and the cell membrane. And so it is the stuff outside the nucleus up to the cell membrane. It's what you usually would think of as composing the majority of the cell. Okay. The cytosol is going to be the fluid part of that cytoplasm. And so it's going to be the aqueous solution of electrolytes and enzymes. It'll catalyze many of the cell's chemical reactions and is the environment that you're going to do all of this in. Okay. This right here is a cell. And so you see the nucleus here, cell membrane here, and then this is the cytoplasm, all that blue part right here. Okay, that's sort of sky blue. By the way, these are different parts of the cell. We're not going to go super into this, but you need to know enough that these are the mitochondria right there. This right here is the mitochondria. The Golgi apparatus is right here. It looks like kind of a long membrane portion. It does a lot of the reactions in your cell. This right here is lysosomes. This is the rough endoplasmic reticulum and ribosomes right there. And ribosomes are where we would doing that, creating that RNA, if you remember. Okay, that's where we were taking the RNA and making proteins. Okay. So once we look inside of the mitochondria, what you'll notice is the mitochondria actually has 2 membranes. We have the outer membrane, and then we have an inner membrane. Okay, and so the outer membrane separates it from the rest of the cell. The inner membrane, on the other hand, is going to be a membrane that's within the mitochondria. And that's because you actually have an extremely, pretty extreme environment in your mitochondria that can be very, very acidic. Okay, very high concentrations of H+. This is actually, in fact, how the mitochondria works. It works a lot like a battery, where you get a high concentration of an ion on one side, and then you let that force that wants to disperse that, diffusion, right? Diffuse those through an energy pathway, which is going to be the oxidative phosphorylation. And that's going to be the driving force that drives ATP synthesis. Okay? So you're kind of building up pressure, and then you're very controlled for releasing it. And that's how your mitochondria actually works. This is called the cristae, by the way. It's these little folds here, in case you're wondering. And this would be the intermembrane space. That's the space between the membranes. So within our cytoplasm, we're going to have specialized structures. These are going to be called organelles. They're going to carry out specific functions of your cell, much like your organs in your body do, right? But you're going to have organelles because they're within the cells. Okay? Ribosomes are going to be your site of your protein syntheses. Mitochondrion is going to be the energy-producing factory of your cell. That's where you've already known that part. And again, you have the outer membrane and inner membrane, and an intermembrane matrix between them. It's referred to as the matrix.
Mitochondria and mitochondria.
Multiple singular. Enzymes located in the matrix along the inner membrane catalyze the oxidation of carbohydrates, fats, and amino acids. Okay, so let's look at the high energy versus low energy forms. So right here's the high energy form for ATP. So this is the adenosine triphosphate. It has 3 phosphates on it. It's a high energy form. When you remove one of these phosphates, this is going to be the lower energy form. Okay? If you were to remove 2 phosphates, that would be the lowest energy form right there. This right here is going to be the NADH. This is the reduced form here. See how it has extra hydrogen on it. So if we go to the oxidized form, it has less hydrogens on it. Can everybody see that okay? Okay. And remember, that was the difference between oxidation and reduction, wasn't it? And so right here we have oxidized form. Here we have the reduced form. By the way, this is that vitamin B3, if I remember correctly. My vision's still a little bit blurry, so correct me if I get that wrong. I'm kind of doing this half off of memory right now. High energy forms, again, are FADH2. This is going to be the reduced form. See the extra hydrogens on them? We get those hydrogens removed, and what we see is the lower energy oxidized form right here. And so that's your oxidized form. And this is how you actually transfer your oxidation reduction states between one place and another. By the way, that's B2, riboflavin, in case you're wondering, right here. So that's why you need a lot of those vitamins, in case you're wondering. And right here, again, we have the acetyl coenzyme A. And right there is your vitamin portion. This right here is the thioester functional group, in case you're wondering. And this right here is the acetyl group over here with the double bond oxygen on it. And so this actually transports these. An interesting part of this should be kind of familiar to you here. Yeah. It's used a lot in all your energy transfer molecules. This is actually the lower energy form right here, where we've actually reduced that thiol right there. And so we no longer have the acetyl group on it. So it's just coenzyme A. All right. So nucleotides are going to act as energy exchangers. They can also be coenzymes, of course, as we've seen in all those examples there. Metabolic nucleotides are going to have your high energy and low energy forms. And so this is going to be putting energy on them and then extracting energy from them. They're primarily going to consist of the nucleoside adenosine, phosphate, and a 5-carbon sugar. So many of these molecules are also going to have a vitamin within their structure, and this is why those are vitamins, and you should get your vitamins, right? Is because it's highly necessary to make these molecules and keep these molecules available. You got it. Yep, to supplement your General Tso's chicken. All right. So ATP and ADP. ATP is going to be your sort of energy currency of your cell. That's the kind of normal way to think about this when we're looking at this in biological context. ATP can undergo hydrolysis to your ADP. During the hydrolysis, you're going to release energy. ATP is going to be a high energy form. ADP is going to be your lower energy form. The chemical given off during the hydrolysis of ATP can again be coupled and drive a chemical reaction that requires energy during anabolism. So what you'll see is oftentimes instead of just you don't just cut the phosphate off and it drifts away. What you do is you actually transfer the phosphate to a molecule, and that makes it higher. Okay? Is that making a little bit more sense? So rarely do you just see it cut phosphate off. That's it. It typically gets transferred to something. It's still a hydrolysis reaction. So the NADH and NAD plus, again, and the FADH, FAD, these are going to be putting hydrogens on and removing hydrogens from. The one with the hydrogens on it is going to be the reduced form. The one with the hydrogens removed from it is going to be the oxidized form. One of the things you will note here is that the naming of these is a little bit loosey-goosey because of all they're really looking at is the hydrogens that change the oxidation state. Okay? And so you may have noticed in like the actual structure for the NAD to NADH, there actually would have been a hydrogen, an extra hydrogen. So like you could have called it NADH2 and NADH plus. Okay. But all they're really caring about is you change the state by 1. So that's why it's called this way. Okay. In case you were noticing that and go, oh, that seems confusing. Now, nicotine mid is going to be derived from the vitamin niacin, which is B3. Riboflavin B2 is going to be found in the FAD. This is where the names come from. So the N here, nicotine amide. And then right here is flavin, right? Riboflavin. Okay, so the acetyl coenzyme A and the coenzyme A is another extremely important energy exchanger. And so the two forms are going to have a difference of an acetyl group. When you have an acetyl group on it, that's the higher energy form. When the acetyl group has been removed, that's going to be the lower energy form. And this is all going to happen by creating a carbon-sulfur bond, and that's going to be your thioester functional group. You're going to hydrolyze that bond, and that's going to give you your coenzyme A, the lower energy form. Coenzyme A is actually going to have an adenosine, 3 phosphates, and a pantothenic acid. Excuse me. Wow. That's vitamin B5. And so next we're going to look at carbohydrates. So we have starch. This is going to be a mixture of amylose and amylopectin. And this is going to be actually begin to be digested in your mouth. And that's by the alpha amylase in your saliva. This is why if you ever have something sweet and then you lick your fingers, it gets really sticky. Okay. So the salivary amylase hydrolyzes some of the alpha glycosidic bonds in your starch molecules. This is going to give you your glucose and your disaccharide maltose and some oligosaccharides as well. Some of them are just going to be broken up into smaller units that are like 3 to 9 usually. Okay. Now, only the monosaccharides are going to be small enough to be transported through the intestinal wall into your bloodstream. The rest of them are going to be too large, and so will actually end up passing through you. This is one of the reasons why certain types of sugars and sugar substitutes can make you need to go to the bathroom really bad. Okay? And that's their main side effect, by the way. case you're ever wondering. The cellulose cannot be digested. We actually lack the enzyme. That's called cellulase. It hydrolyzes a beta glycosidic bond. We do not have that enzyme. So this right here is actually your pathway. So you have your polysaccharide in your mouth. You just ate something starchy like potato. And then you're going to have your alpha amylase in your saliva. It's going to break it up into these things: oligosaccharide, maltose, glucose. Your glucose can go directly into the pathways, which will be glycolysis. The maltose actually needs to go down here and get broken up along with the oligosaccharides. So right here is the oligosaccharides. The pancreatic amylase is going to break those up into typically maltose. Maltose is going to then be reacted using maltase and is going to be split up into its 2 glucose units. By the way, if you have a functional lactase, you're going to take lactose and you're going to break it up into galactose and glucose, if you're wondering. And this right here is sucrose. That would be sucrase. It breaks it up into glucose and fructose. So once you have these broken up into monosaccharides, these can absorb through your intestinal walls, and now you can actually bring those across into your bloodstream. So once they get into the bloodstream, they can make their way to your cells, and that's where we're going to start breaking them up into, you'll have glucose, and it'll start being broken up into the pyruvates that we had mentioned before by glycolysis. Okay, so let's talk about breaking up fats. So dietary fats are going to be your nonpolar molecules. They're going to assist in digestion. We actually need something to move these around. And so we actually have something called bile. This is going to be excreted from your gallbladder into the duodenum, the upper portion of the small intestine, during digestion. Damn, it's killing me. So your bile is going to contain amphipathic molecules. These are referred to as bile salts. Basically, it's going to be molecules that act a lot like soap. Okay? And it will make a small shell that will be water soluble around these so that they can be transported into your aqueous environment, which is your blood, right? And so the amphipathic bile salts will place their nonpolar face towards the dietary fats, their polar face towards the water, and they're going to form those micelles, just like we had referred to when we talked about soap working, right? And so they're going to break up these large nonpolar globules into smaller droplets, which are the micelles again. And again, that's going to be emulsification, just like soap works, okay? They're just slightly different looking than soap. All right, now, again, your smaller micelles are going to move the dietary fats closer to the intestinal cell walls. Cholesterol can then be absorbed, and triglycerides can be hydrolyzed into free fatty acids and monoglycerides. Once you break these apart, they're going to be small enough to actually be able to be absorbed across your cell membranes. So once the intestinal wall, you have your free fatty acids and monoglycerides. Once past that intestinal wall, you're going to reassemble those in your triglycerides again. Your cholesterol is going to be linked to another free fatty acid. This is going to form your cholesterol ester. And you're going to repackage that as a lipoprotein that's called a chylomicron. Okay. The chylomicrons are going to ultimately be what transports the triglycerides to the tissues, where they're going to be used as energy production, or you can store them in cells, one of the two, or you can modify them further to use them for things like cell membranes, etc. Okay. Those are going to be your chylomicrons. So right here we have our small intestine with our fatty acid, triglyceride. And so we go ahead and start getting that into your packaging to get close to your small intestine. Once it's close to the intestinal wall, we can actually start reacting these. So what we're going to do is we have this package with cholesterol. We're going to have the pancreatic lipase here, and it's going to remove fatty acids. Once we've removed these fatty acids, we're going to have this absorb through the intestinal wall, and then we're going to reassemble them. And so we reassemble that back into the triglyceride. That's going to be joined together with a protein, and that's going to give us chylomicron. By the way, this right here is the cholesterol with the free fatty acids that's going to give us our cholesterol ester that's required to make these chylomicrons. Once we have in the form of a chylomicron, this can actually go through our bloodstream. It's going to be swollen in our bloodstream. It can move around, and we can absorb it into tissues and our different cells. Okay, so proteins. These are very large, obviously. By themselves, they're not going to be able to go through the cell membranes, and so they're going to be just too large to do anything. So the first part of this is protein digestion is actually going to begin in your stomach. And so your proteins are going to be denatured, unfolded, using your acidic digestive juices. Your digestive enzymes like pepsin, trypsin, chymotrypsin are going to hydrolyze those peptide bonds. So once you've unraveled them, you're going to start chopping them up. The amino acids are then going to be absorbed into the bloodstream for delivery to your tissues. And so now we have them in our bloodstream, and we have them going to our tissues and our cells ultimately. So our main source of fuel for the body is actually going to be glucose. This is going to be catabolized through chemical pathway called glycolysis. Glycolysis cutting up glucose. When there's not enough glucose entering the body during periods of fasting or sleeping, your body is actually going to make its own glucose. This is through an anabolic process, and this is gluconeogenesis. Notice gluco as in glucose, right? Neo, new. Genesis, making. Okay? So making new glucose. Glucose is going to enter your cell from the bloodstream through a glucose transporter protein in your cell membrane. Inside your cell, glycolysis occurs in the lipid portion of the cytoplasm, or the cytosol. When the 6-carbon monosaccharide glucose is broken down into 2 3-carbon molecules of pyruvate. Notice I mentioned we're going to end up at pyruvate a lot, right? And so we're going to break the glucose down into pyruvate. The other hydrolysis products can also be used as fuel and are going to give you a bit of that energy as well. So in the body, energy must be transferred in small amounts to minimize heat released in the product. This is why you don't just like burn these things. If you were to do burning, you would light on fire, right? You wouldn't actually function. You die pretty quickly. And so the reactions that produce energy are going to be coupled with reactions that acquire energy directly. So you have minimal energy waste in the form of heat. And this is going to help to maintain a constant body temperature because you're just going to be generating small increments of heat, not large amounts. So in glycolysis, energy transferred through the phosphate groups is going to undergo condensation and hydrolysis reactions. There's going to be 10 chemical reactions in glycolysis that result in the formation of 2 molecules of pyruvate from 1 molecule of glucose. So this is going to be the overall chemical reactions of glycolysis here. So right here we see glucose. We're going to have 2 NAD pluses. Remember, that's that oxidized form, low energy. We're going to have 2 ADPs, again, the low energy, right, along with their 2 phosphates. This is then going to give us pyruvate, 2 of those. We're going to get 2 NADHs. Remember, NADH is that high energy form, right? The reduced form. This is the H plus that would come with those. Remember how I was talking about it can be referred to as NADH2? These are not really worth thinking about that much, okay? But it makes 2 H pluses. It also makes 2 ATPs. So we've made 2 of the energy molecules at this part, okay? All right, now, the first 5 reactions are going to require energy investments of 2 molecules of ATP. So when we do this reaction, remember, this is the overall thing. This is actually going to be split up through a series of reactions, which is our pathway, right? During this pathway, we actually have to set it up first. Okay. And that setup is going to require 2 ATPs. And so this is actually one of the reasons. You ever eaten a bunch of stuff and then you feel tired right after? This is one of the reasons. It takes energy to start it. And so you end up actually like losing energy when you first start. This is why if anybody's ever starving to death, you don't just like feed them regular food, a bunch of it. You feed them just a little bit, right? Because you can actually take up so much energy, they just fail. So the molecule is going to be split into 2 sugar phosphates, and that's that first part of this reaction. Reactions 6 through 10 are going to generate 2 high energy NADHs. This is where you get your payback. You get energy back out, and 4 ATP molecules, which means net, we make 2 ATP, right? That's our net pay for this. The 4 phosphates are going to be removed from the sugar phosphates, by the way. This right here is glycolysis. This is the first part right here. So we're going to have our glucose. We're going to use an ATP to set it up, and this is going to generate our glucose 6-phosphate. Notice we attach a phosphate on there. Okay? That makes this molecule higher energy. And so now the next step, we're going to do a rearrangement, and this is going to give us fructose 6-phosphate. We're then going to attach another ATP, another phosphate on there. This is going to give us a mostly symmetric structure, 2 kind of equal portions that we can break apart. Okay? And so that's the fructose 1,6-biphosphate. What happens next is it actually gets broken apart, and it actually ends up generating 2 molecules here that are slightly different. But there's actually a pathway to rearrange this one right here, which is the dihydroxyacetone phosphate, to this other molecule. So what you'll end up with is 2 of these molecules here at the end of this. And that's going to be glyceraldehyde 3-phosphate. Okay? So you got 2 of those so far. That's what you end up with this first page. Now, the next part of glycolysis, what you're going to see here is this is the energy generation phase. So what we're going to have here is our 2 phosphates, our NAD plus is going to go to the NADH right here, those 2 NADHs. Remember, each of these steps we're going to do twice, right? So we're doing it for one of these molecules and then the other. That's why we get 2 here, because we did it twice. This is going to give us our 1,3-biphosphoglycerate. Notice we've just added a phosphate to the other end of this one. This is a very high energy molecule, and so we can go ahead and harvest some of that ATP off of here. Okay? And so what we're going to get here is we're going to do this twice, each generating an ATP from an ADP. This is going to give us our 3-phosphoglycerate. We're going to have this rearrange. This is going to give us the 2-phosphoglycerate. So we rearrange that to have phosphate in a central portion of it. It also gives us 2 waters for the next part. So we're going to, again, do this next process where we're going to go ahead and do a condensation where we remove the waters. And this is going to be a phosphoenol pyruvate. Notice we're getting closer and closer to pyruvate, aren't we? Okay? And that's the point here. These are rearrangements to make this pyruvate. Okay? And so we're going to go ahead and harvest that second phosphate here. That's going to give us our 2 more ATP. Notice that we've paid ourselves back in this first one. And now we've generated an extra 2 ATP. Okay? So at this point, we've generated a bit of net energy. All right, that will give us 2 pyruvates. These pyruvates can then go on into the mitochondria, and they can go on to the next portion of this. Okay, so let's go ahead and talk about these are the steps that we just had gone through. Notice again, this was that first part where we invest that first ATP, right? This right here is that rearrangement. This is called an isomerization because we're going to form isomers of it, right? Different arrangements in space. Okay, so we have a rearrangement. This is called phosphorylation because we added a phosphate to it. So we put a phosphate on. All right. The next part, we're going to use our second ATP investment. This is going to give us that phosphorylation again. And this is where we get the, see how it's almost symmetric? Not quite, because one is up, one is down. These are slightly different, right? OH here, but almost symmetric, isn't it? A lot more symmetric than the glucose was. Okay. And so the next part is going to be an isomerization, where we do another rearrangement here. Oops, excuse me. Okay. And so once we have that nearly symmetric form, which is the fructose 1,6-biphosphate, we're going to do a cleavage. And then because they're not quite the exact same molecule, we don't want to have two different pathways, all the enzymes that would take, etc., right? You want to keep it simple. And so what's going to happen is then we're going to do another isomerization, and that's actually going to be for a triose. If you didn't realize this right here was a triose, wasn't it? It was a sugar that's 3 carbons. Okay. Okay. And so we split, we get these 2 forms here, and then we're going to rearrange this so it's that. So that we have the exact same thing twice. Okay. Once we have these glyceraldehyde 3-phosphates, 2 of those, this is where we're going to start regaining that energy. Okay. So we're going to start producing energy. The first thing we're actually going to do here is we're going to do an oxidation. And so this is going to give us our reduced NADH. And we're going to oxidize this glyceraldehyde 3-phosphate here. Okay. And that's this right here. See, it has less hydrogens, more oxygens now. And that's oxidized right there. And so that's going to give us the 1,3-biphosphoglycerate. The next thing we're going to do to that is we're going to actually get our second ATP out of this. We're going to start harvesting those phosphates off it. Okay. And so we harvest this phosphate off of it right here, and that's going to give us our ATP. So we're finally getting ATPs back. Right? We've paid ourselves back now. The next part, we're going to do again the formation of the 2-phosphoglycerate. Notice how this is just going to be a rearrangement right there. So now we've rearranged, we've set ourselves up, and we're going to have the removal of water here across the structure. So notice we remove the water here, a hydrogen here, and an OH here to form the double bond here. Okay, so that's going to be our phosphoenol pyruvate. By the way, that enol part right here means double bond next to an alcohol. Okay. So the next step is going to be removing that last phosphate. So once we chop off that last phosphate, we're going to get another 2 ATP overall. Notice that's 1 ATP per cycle of this, right? 1 ATP per one of these molecules. But we made 2 from the glucose, excuse me. Excuse me. And so our pyruvate here is going to be produced. We're going to produce 2 of those. Now let's talk about regulation of your glycolysis. So the main step of regulation in glycolysis is actually step 3. Too far. Right there. That's your main regulation step. Okay. And so that's going to be the point where you're going to regulate this, and you typically won't let this go any further if you don't need more energy from this. Okay. Notice it's at a step where you'd have to input more energy, isn't it? So you can minimize your energy you put into this. Now, the enzyme phosphofructokinase is going to catalyze that phosphorylation of fructose 6-phosphate to the fructose 1,6-biphosphate, that second phosphorylation. It's going to heavily regulated by your cells. And the ATP is actually going to act as an inhibitor of the phosphofructokinase. So if I already have enough ATP, I'm not going to go forward. I'm just going to leave it as that the fructose 6-phosphate. I'm not going to go any further forward. Because that fructose 6-phosphate actually has a bunch of pathways it can go in and do other stuff. Okay? I can make other things from that. So if you have plenty of ATP, what's going to happen is your glycolysis is going to slow down, and you're not going to chop up your glucose as much, and you're going to keep more of that just in the glucose or the fructose 6-phosphate forms. So let's talk about the fates of pyruvate. Your metabolite, pyruvate, is going to be generated from glycolysis. This is going to be catabolized further, and it's going to produce a lot more energy for the body. This second part is actually where you get the majority of the energy per molecule. Your fate of your pyruvate is going to depend on the availability of oxygen in your cell. So when you have ample oxygen, this is going to be referred to as aerobic conditions. Is everybody comfortable with that? Aerobic meaning oxygen, anaerobic meaning no oxygen. Pyruvate is going to be oxidized further to the acetyl coenzyme A. So remember where we were talking about the acetyl coenzyme A's. You're going to oxidize the pyruvate to those. When oxygen is in short supply, again, those are anaerobic conditions, your pyruvate is going to be reduced to the lactate. And this is quite inefficient, by the way. This is also why you get lactic acid buildup. Do you guys know when you get lactic acid buildup? When you exercise a lot, right? What happens when you exercise and they're doing really, running really fast or whatever, exercising really hard? Well, your tissues are going to start depleting and running low on oxygen, right? This is why when you're exercising really hard, you start breathing really heavy, right? You're trying to get more oxygen because you're in anaerobic conditions. So that's actually lactic acid buildup, in case you're wondering. Now, when oxygen is readily available in your cell, your pyruvate is going to produce more energy for your cell. When the pyruvate breaks down further, the carboxylate functional groups of the pyruvate, these are going to be liberated as CO2. This is called oxidative decarboxylation. The oxidative is actually referring to the oxidation reduction reaction part of this, that the CO2 is the oxidated form, right? The metabolite acetyl coenzyme A is actually going to be formed. Your reaction is going to occur in your mitochondria. So under anaerobic conditions, your middle carbonyl in the pyruvate is actually going to be reduced. And so we're going to add a hydrogen to that. And we're going to get an alcohol functional group. And that's going to give you your lactate. And so the hydrogen and the energy required for this reaction is actually going to be supplied by NADH and H pluses that we saw from the previous reaction. This is going to produce NAD plus. Note that no ATP is generated by the pyruvate under anaerobic conditions. Your NAD plus is actually produced, is going to funnel back into glycolysis. This is going to allow you to oxidize more glyceraldehyde 3-phosphate, and you're going to produce those small amounts to each cycle of ATP, right? And so realistically, what this is you tend to run out of the NAD pluses pretty easily because they're fairly large molecules. They're meant to be recycled. And so if you can't go forward with your normal metabolic pathway using oxygen, you start freeing those up so you can just run glycolysis a bunch. Okay? It's really, really inefficient. But if you don't have any oxygen, it's better than nothing. Right? So again, this reaction is actually going to occur in the cytoplasm. Yeah, super inefficient though. So this right here is just a summary of what I just talked about. And so right here we have the pyruvates so far. And so aerobic conditions, we're going to go off into the citric acid cycle, going through acetyl-CoA. If we're under anaerobic conditions in our cytoplasm, we'll free up that NAD+. That will give us lactate. And then we can put the NAD plus back into a glycolysis. And we can continue running these reactions. Okay. So yeast is actually going to convert pyruvate to ethanol under anaerobic conditions. This is why like brewer's yeast does brewing. You all are aware of that, right? Yeah. Okay. This process is called fermentation. And so the preparation of alcoholic beverages, of course, yeast produces pyruvate from glucose, and like grape juices, any sugary substance or starchy substance that can work. You have to start breaking apart the starch to let it run better. And so under these low oxygen conditions, it's going to transform pyruvate into ethanol. And there we get pyruvate with NADH transforming into ethanol and CO2. This is in fact why the like beer, if you see it, has ethanol and then it has CO2. That's actually the fizzy, the carbonation of beer is that production inside of that vat of the CO2 makes it carbonated. Okay. It doesn't actually have to be externally carbonated, though they usually do now to make it fizzier. Okay. By the way, this is the waste product for this. It is poisonous to the yeast. And so the yeast actually will kill themselves off after typically about 4-ish% concentration ethanol. Okay. And so that's kind of the limit of most wines and beers through fermentation because it kills the yeast off because they're living in their own waste product. All right. So fructose and glucose and glycolysis. So fructose is actually readily taken up in the muscle and liver. In your muscles, it'll actually be converted to fructose 6-phosphate. And so the fructose just straight up slots into the fructose 6-phosphate portion of glycolysis. Okay? And that's how you handle fructose. Note that step 3 in the liver, it can actually be converted to trioses that can be used in step 5. So if it makes it to the liver, that's actually the step it'll join. Fructose that enters a cell is going to flow from steps 5 to 10 in glycolysis. Fructose uptaken by cells is not regulated by insulin. So all fructose in the bloodstream is actually forced into catabolism. Glycolysis is actually going to be regulated at step 3. The triose products created in the liver provide an excess of reactants that create excess pyruvate and acetyl-CoA. If not required by energies in the cell, it's actually going to be converted to fat. And so this is actually why you can oftentimes see like in a very, very high fructose diet, very, very high sugar diets, you can actually see a lot of lipids around your liver. This is part of the reason. All right. So again, these are the regulation steps right here of your glycolysis reaction. And so glucose is going to be regulated through your glycolysis reaction. Your unregulated fructose is actually going to enter glycolysis typically over here in the step 5-ish. This is going to give you excess product is going to be converting to fats right here from your glycolysis products. If you have excess of these and you don't have enough usage of them, that's going to give you your fats. This is why even if you don't eat any fat, you can still gain fat. You still make fats from your products from glycolysis. Okay. Anybody know how much sugar you actually need in your diet? 0. There are no essential sugars. Okay. You can make all sugars that you need. Yeah. So you actually don't need any sugars in your diet whatsoever. Okay. There's a few fats that you need, not that much though. And obviously there's a few proteins you need as well. Okay. Or I should say amino acids, excuse me. Now, the citric acid cycle, this actually goes by a tremendous number of names, but typically you'll hear citric acid cycle or Krebs cycle. This is going to be a series of reactions. It's going to degrade the 2-carbon acetyl groups that we got from those previous metabolic pathways into the metabolite acetyl-CoA. This is going to transform that into CO2 ultimately, and we're going to generate NADH and FADH2. Now, one of the things that I need you to kind of understand here is the citrus acid cycle is in the mitochondria, and it is going to be directly linked to the other cycles I'm going to mention, okay? So we're going to have the electron transport chain and the oxidative phosphorylation. Everybody kind of remember those? Okay. Now, the citric acid cycle is directly intermeshed and linked to those. They're not independent things. Okay? They're kind of like your arm and your body, right? They work together to do things. Okay? You don't really have one without the other. All right. So, that citric acid cycle actually originated because the first step in the molecule involves the formation of a 6-carbon molecule that's called citrate. And so it's referred to as the citric acid cycle from that. That formation of the citrate is actually what starts the cycle. It's going to be in a condensation reaction between the entering acetyl-CoA molecule and your 4-carbon oxaloacetate molecule. This is actually what makes it in a cycle because you always regenerate the oxaloacetate and then you add an acetyl. And you just keep doing that over and over. New acetyls each time. Okay? So this right here is the citric acid cycle. So we have our oxaloacetate, which was part of the last molecule that went through this. This is going to join with an acetyl-CoA. You're just going to take the acetyl group and transfer it off onto the oxaloacetate. This is going to give you citrate. The next part, we're going to generate CO2 and NADH. This is going to give us our alpha-ketoglutarate. Notice that this was actually this carbon here, wasn't it? Not the new acetyl-CoA, that new acetyl group. Okay. So, the next step, you're going to go ahead and again generate a CO2 and an NADH. This is going to give you succinyl-CoA. What I'd like to point out again is your acetyl group you just joined in, again, is not the source of the CO2. Okay? We're actually chopping off the whole part from the previous cycle. Okay? Now, the next step that's going to happen is this is going to then generate GTP. Notice how I said, we're going to have ATP, and GTP is sometimes that energy. That's where the GTP is generated. Instead. It's just a kind of interesting little side note to this, is you actually end up generating GTP instead of ATP here. Okay. Notice you got a lot less ATP in this cycle than you would have thought, right? You probably thought this was going to generate energy, didn't you? Well, it does, but it does it in the form of NADHs and FADH2s, doesn't it? Okay. So we actually need a cycle later that's going to use those to generate ATP, don't we? So this generates your GTP, and then we have another step that's going to generate the NADH2, and then another step that's going to generate the NADH, and we'll get oxaloacetate. And then we start the cycle anew. Okay? Notice that these steps right here essentially are just rearrangement steps, oxidation reduction steps. We don't really change the number of carbons. Okay? So that CoA is 2 carbons from the original structure and 2 carbons from the acetyl group are what give you your final oxaloacetate. Okay? And then we're always kind of chopping the ends off of that. So let's talk a bit about those reactions. We have reaction 1. This is going to be your formation of your citrate. So the acetyl group from the acetyl-CoA, which is 2 carbons, will combine with the oxaloacetate, just 4 carbons. It's going to give you your citrate, which is 6 carbons, and CoA, coenzyme A. Notice that these coenzyme A's are always going to end up being regenerated. Okay? We're always going to end up freeing them back up at the end. The reaction 2 is going to be your isomerization to isocitrate. So you're actually going to make isocitrate from citrate, literally just an isomer rearrangement of citrate. The OH and one of the H atoms are going to be swapped in the citrate, so you form the isocitrate. This is actually necessary rearrangement because the isocitrate is going to be oxidized in your next reaction. that OH wouldn't be available to oxidize at the same sort of energies unless you rearrange it. So it's actually vital. Reaction 3, you're going to have your first oxidative decarboxylation. That's your release of CO2. One of the things I'd like to point out is these CO2 release steps, they are actually irreversible steps. You can't actually go backwards from those. Okay? So whenever you release a CO2, it's going to diffuse and go away from your cell. And so you can't go back from those. Those are commitments. You're done. You're going onwards. Okay. Irreversible steps. Now, that isocitrate is going to undergo an oxidation. You're going to have 2 hydrogens that are removed to a ketone. This is going to be called alpha ketoglutarate. And your NAD plus is going to reduce to NADH, which accepts the proton and the electrons removed from that oxidation. And so this is going to be where you are reduced, which accepts the electron density from the oxidation. The 6-carbon isocitrate is decarboxylated. This is going to give you a 5-carbon then, because there's 1 carbon in CO2, right? And so that's then going to give you alpha ketoglutarate. Reaction 4, your second oxidative decarboxylation. So your thiol group of the coenzyme A is going to be oxidized, which is losing a hydrogen. And another NAD plus is going to be reduced, NADH. The alpha ketoglutarate, that's the 5-carbon one, is going to be decarboxylated into succinyl group. It's going to be a group because it's attached to CoA now. Okay? So it's going to be the succinyl group attached to the coenzyme A. And again, that CoA is going to be bonded to that succinyl group. And so it's going to be succinyl CoA. Reaction 5 is going to be a hydrolysis of the succinyl CoA. And so this is going to undergo a hydrolysis. It's going to give you succinate and coenzyme A. Notice you've regenerated coenzyme A. The energy produced is going to produce the high energy nucleotide guanosine triphosphate, which is your GTP. And so this is going to be generated from your GDP, just like an ATP is generated from an ADP, and a free phosphate group. Your GTP, by the way, can be transformed in your cell to ATP. Like these things can all be transformed back and forth. So like if you have a D, the deoxy one from DNA, right? DATP, it can be transformed into an ATP. You can just move those phosphates and the energy over. And same thing can happen to a GTP, a ZTP, TTP, whatever. Okay? You can interconvert those. It takes a little bit of effort and energy, but it is something that your cell does and needs to do quite a bit because you need to make new DNA all the time, right? So, an important pathway in your body. Now, your reactions, again, reaction 6 is going to be dehydrogenation of your succinate. So 1 hydrogen is going to be eliminated from each of the 2 central carbons of your succinate. This is going to give you a trans carbon-carbon double bond, and it's going to produce fumarate. The 2 hydrogens will be reducing the coenzyme FAD to FADH2. And then reaction 7 is going to be your hydration of fumarate. So your water is going to be added across that trans double bond. It's going to give you a hydrogen and an OH on the other side, and that's going to be malate. Now, the next step is going to be, and the final one, is going to be the oxidation of malate. So in reaction, just like in reaction 3, your secondary alcohol of the malate is going to be oxidized to your ketone. This is going to regenerate oxaloacetate, so you can go another cycle. And this is going to give you your protons for the coenzyme NAD+, to give you NADH. This is the complete cycle with all the steps in it and all the structures that I just mentioned. Okay? And so that's the whole thing. All right. This right here is a summary of it. And so again, it's going to be an 8-step process. You'll have 2 CO2s that are generated. 1 coenzyme A is going to be produced, i.e. liberated from this process. It comes in, and it comes back out. Okay. One turn of your citric acid cycle is going to produce net energy output of 3 NADHs, 1 FADH2, and 1 GTP, which can be converted to an ATP and is energetically analogous to an ATP. Your net reaction for one turn of this 8-step cycle again is an acetyl-CoA, 3 NAD pluses, a FAD, GDP, and 2 waters, gives you 2 CO2s, 3 NADHs, 2 H pluses, an FADH2, coenzyme A is regenerated, and a GTP. Okay. Now, 2 ATP are produced in glycolysis, 2 ATP are produced in the citric acid cycle, and we haven't really made that much energy, have we? Sounds spectacularly unimpressive, doesn't it? Okay. And that's because we're not thinking of the energy in NADH and FADH2 yet. We haven't used those yet to produce ATP. And so the NADH and FADH that are produced in glycolysis is 2 NADH per glucose. From pyruvate oxidation, it's going to the acetyl-CoA is going to give you 2 NADHs per glucose again. And then the citric acid cycle, we're going to get 6 NADHs and 2 FADH2s per glucose. Has anybody noticed that we haven't actually used any oxygen yet? There's no oxygen on this side, is there? No. Yeah. Yeah, it's just the H2 water, right? Got a lot of water in you. You're mostly water, aren't you? So, it's because we haven't gotten to that part yet. We need the oxidative phosphorylation. So the high energy reduced forms of your nucleotides are going to transfer electrons and hydrogens through inner mitochondrial membrane. This is going to form water. The energy generated as a result of the process is going to be used to drive a reaction that's going to transform ADP to ATP. And that's going to be oxidative phosphorylation. Okay. So we haven't gotten one part of this yet, and that's what connects these two things. So we have oxidative phosphorylation that's going to be driven by those NADHs and FADH2s, right? And that mechanism that's going to build up charge to drive that oxidative phosphorylation, that's the electron transport, okay? Now, again, I know that I'm talking about these things like they're like things you plug in one after another, okay? But that's because if I went back and forth on these, it wouldn't make any sense. And so what I have to do is basically I'm describing one cog right here, and I'm giving you the other cog now, okay? And so that citric acid cycle, it's going to be like a cog that's going to move, and it's going to power the other part to make stuff, okay? They're very interlinked. So the mitochondria, again, are going to be the ATP factories of the cell. The reduced nucleotides from the citric acid cycle are going to be produced there. The energy upon oxidation is going to be used to generate ATP. The reaction of the citric acid cycle is going to occur in the matrix of the mitochondria. And the NADH and FADH2s are going to begin their journey through the inner membrane here. And so currently you have these things on one side of that membrane we've been talking about. Okay, so what we're going to have next is we're going to have enzyme complexes. These are going to be embedded into those inner membrane of that mitochondria. And so we're going to have enzyme complexes 1 through 4. These are embedded in that inner membrane of the mitochondria, and that membrane is going to contain a set of electron carriers that's going to transport the electrons and protons, of the NADH and FADH2 through the inner mitochondrial membrane. Remember, the membranes are nonpolar, right? And so H plus can't normally pass through them, okay? So what we're going to do here is this entire process we've been talking about, it's all going to be for building up a reservoir of H plus inside of that inner mitochondrial membrane. That reservoir of H plus is going to give you this energy source, kind of like a charged up battery. Okay. Or if you were to pressurize a bunch of like water in a tank, right? That would all give you energy that you can drive things with. And this is going to be what drives that next process, which would be the oxidative phosphorylation to make ATP. So 2 of the electron carriers, which are coenzyme Q and cytochrome C, are not going to be firmly attached to any one complex. They're actually going to be used to shuttle electrons between the complexes, and they're going to be lipid soluble, so they can actually move in that membrane. Okay? And so they're going to be referred to as shuttles. This right here is your inner membrane space right there. Right there is your matrix. And so that's your mitochondrial matrix. And this is the inner mitochondrial membrane. This structure is typically located on those little parts that jut out on the mitochondrial membranes, okay? And so what you'll have here is you have your NADH going to NAD+. Remember that NADH we made first? That's going to come in here. And this is going to help us transport an H+ across this membrane. And it's going to do so by using the electron to move it along, okay? So what there are is basically there's little structures in here that the electrons can hop off onto. And the electron moving is going to pull the H+ with it, right? And so you're going to pull that H+ through. This is the pathway of the electrons, by the way, that we're going to look at. Okay, so complex 1 is going to get an electron loaded on it. This is going to be transferred to Q here. Remember how it's talking about coenzyme Q and cytochrome C? That's the coenzyme Q right there. Okay, complex 2 then is going to be using FADH2, going to FAD, to move a hydrogen, H+, and an electron onto complex 2, which will be moved onto Q and put into complex 3. Okay. So these are powering complex 3 so far. Complex 3 is going to readjust essentially. It's going to be like your transformer near your house, right? It's going to readjust things. And then cytochrome C is going to be what takes up the electron from complex 3. Complex 4 is then where you're going to actually have these electrons that are going to actually be used, and this charge actually get used in this process. And this is where you're going to actually use up your oxygen. See your oxygen here? This is finally where we use the oxygens. And we're going to generate water. You all probably thought the oxygens and CO2s were directly correlated, didn't you? They're not. Okay? The CO2s are generated in the citric acid cycle. You actually are moving the charges through NADH and FADH2. And those charges are what are then using the oxygen. Okay? And that's in the form of H+ and electrons. Okay. So let's talk a bit more specifically about the complex 1. Actually, we're going to stop here, right? So this is the thing I want in your head for this next part. Okay? Everybody have a good break?
We had a really long test today. Really spent most of it doing.
Oh my goodness.
I